Welcome back everyone, I am Saptaparni from BioLive. Hope all of you are doing very well. So in today's video we are going to discuss about genetic crosses in Drosophila. And today we will be mainly focusing on 50% male lethality crosses for Carli mutation. Okay, so in my previous 3 videos I have already discussed about eye color mutation, then vestigial wing mutation and Carli wing mutation in Drosophila and related genetic crosses. From now onwards, we will be focusing on lethality crosses. Today, we will focus on 50% male lethality, then 100% male lethality, then 50% female lethality, then 100% female lethality, and so on. Okay. So, before starting today's video, I would like to request you to kindly subscribe to my channel for more such videos, and please like the video if you really like it and it becomes useful to you. Okay, and don't forget to share it with your friends. So without further delay, let's begin. So in case of 50% male lethality, what should we check? How will we understand that 50% male lethality is occurring? See, till my previous 3 videos, we have seen that the sex ratio, that is male is to female ratio, was 1:1. But whenever there will be lethality, 50% male lethality or 100% male lethality or female lethality, whatever, the ratio will not remain same. The ratio will not remain 1:1. For example, in case of 50% male lethality, the ratio becomes 1:2. Where you can easily see that for one male there are 2 females. That means 50% males are died. Okay, 50% males are dead. Okay, here. So as today we are going to discuss about Carli mutation and related 50% male lethality, we must also know some facts about Carli mutation. For that, I would like to request you to kindly visit my Drosophila genetic crosses in Drosophila for Carli mutation video. I will give the link below in the description box, and I will also attach it at the end of the video. Okay. So what are the 2 major factors you must remember while learning about Carli mutation? See, Carli, which is denoted as CY allele, is a recessive lethal allele. Okay, what does this mean? This means when there will be homozygous condition, like there will be 2 CY alleles on the second chromosomes, homologous chromosome of Drosophila, that progeny will be dead. Okay, it will be lethal. Okay, but you have to remember that phenotypically this Carli mutation is dominant over wild type wing allele, that is denoted as CY plus. Okay, so it is a autosomal dominant mutation. Okay, this Carli mutation gene is present in second chromosome, that is why it is a autosomal mutation, and Carli mutation allele or Carli mutant allele is dominant over wild type wing allele, that is CY plus. Here you can see the Carli mutant allele CY is dominant over CY plus, which is wild type of wing allele. Okay. So obviously as CY or Carli mutant allele is dominant, that means in both homozygous and heterozygous condition it should show Carli wing. But you can see only in heterozygous condition, that is CY plus or CY plus CY, it is showing Carli wing. Whereas as this Carli mutant allele is also recessive lethal, that is why in homozygous condition, that is CY, it is lethal. Okay, it is not showing Carli wing, rather it is lethal. The progenies are dead here. Okay. On the other hand, as the CY plus or the wild type wing allele is recessive, that means only in homozygous condition it will show wild type of wing in Drosophila. So only when both the second chromosome of the Drosophila will contain CY plus or wild type allele, then only wild type wing will be formed. Okay, so this is the wild type wing and this is the Carli wing, which is called backwardly. Okay, got it? So now let's dig into the questions. See here, the first question says these are the F1 generation progeny data, their phenotype and their respective numbers. So from this you have to derive the P generation cross or parental generation cross. Okay. So how we will do that? See, for any cross, we must derive the phenotypic ratio and sex ratio. So what will be the phenotypic ratio here? Curly is to wild will be, how many curly winged flies are there? 12 plus 6, 18. Okay. And here also how many wild type flies are there? 6 plus 3, 9. So the ratio is 2:1. You have to remember that as the curly is a recessive lethal allele, for recessive lethal alleles the monohybrid cross ratio is 2:1 instead of 3:1. So it is a monohybrid cross. Okay, first thing first. Then let's derive the sex ratio from here. How many males are there? See how many males are there? 6 plus 3, 9 males are there. Okay, and how many females are there? 12 plus 6, 18 females are there. Okay, so what will be the sex ratio? The sex ratio will be 1:2. That means for 2 female individuals there is only 1 male individual. That means 50% of male progenies in the F1 generation is dead. Okay, so it is showing that this data is from 50% male lethality data. Okay. So from these we have to derive the cross. See here, as we can see that it is carrying the monohybrid cross ratio, that is 2:1. From my previous video, if you have watched those videos, you know that in case of monohybrid cross, we consider both the parents as heterozygotes. Okay, so in P generation we are taking mother as heterozygote. So one chromosome, second chromosome will carry CY or mutant allele, and another chromosome will carry wild type allele CY plus. Okay, and in father also we are taking heterozygote, that is CY and CY plus for the second chromosome. Okay, now these are the X chromosomes for mother and these are the X and Y chromosome for father. In our previous video we have seen that in case of father and mother, X chromosomes and X and Y chromosomes are of wild type. Okay, but here lethality has occurred. That means 50% of the males are dead here. Okay, so you have to introduce a lethal allele in the sex chromosome. Okay, now this lethal allele is denoted as small l. So small l is the lethal allele that is present on the X chromosome. Okay, now where should we put this small l allele? So whenever small l allele will be present, you have to remember that it is a recessive allele. Okay, which is recessive under L plus. When L plus is there, no death will be occurring, but when L will be present in homozygous condition, then death can occur. Okay, so if we put L on father here, if we put L on here, so father will itself get dead. Okay, so we cannot put L here. So we must put L plus in the father's X chromosome. Okay, now for mother too, we can put L on both the X chromosome. Okay, but we will not put that, because if both the X chromosome carries X, then in homozygous recessive condition mother will also get dead. So we cannot put L on both the X chromosome. But we need to kill the F1 generation males. Okay, so we must introduce a L allele here. So the L allele will be introduced on one X chromosome of the mother, whereas other X chromosome of the mother will carry L plus, and the mother will be viable. Why? Because L plus is dominant over L, so in heterozygous condition the mother will be viable. Okay, got it? So from gametes, so what gametes can we got from here? See here. Okay, before getting the gametes, you must know that whatever male in the F1 generation will get this allele from mother and this Y chromosome from father, those males will be dead. Okay, because males have only one X chromosome, and if that X chromosome which is derived from her mother is carrying L allele, that is the lethal allele, then obviously the male will not be viable. Okay, it will be dead. Okay, got it? And that is how 50% male will be dead here. So let's see the crosses. See here what gametes are here, can be formed from the female or mother. Obviously one will be CY with L plus, okay, CY with L plus. Another one will be CY with L, CY with L. Then another one will be CY plus with L plus, CY plus with L plus, and another one will be CY plus with L, CY plus with L. Okay. Similarly, from father, one gamete will be CY with L plus, okay, CY with L plus. Another one will be CY with Y chromosome, CY with Y chromosome. Then CY plus with L plus, CY plus with L plus carrying X chromosome, and CY plus with Y chromosome, CY plus with Y chromosome. Got it? So now we are doing the checkerboard here, where I have written all the male gametes over here and all the female gametes over here. Okay, got it? So when we have done the checkerboard, we can see that the first individual is carrying CY. We know that whenever both the autosomes are carrying CY, CY is itself a recessive lethal allele. So obviously it will be dead. Okay, so it is lethal. Got it? This one is also carrying CY, so it is also dead or lethal. Okay. Now it is carrying CY plus, that means it is Carly. As we know, CY plus is dominant over wild type of allele CY plus, so it is Carly, and as it has 2 X chromosomes, so it is female. Okay, Carly female. Now it is also CY plus, that means it is Carly, and as it has 1 X chromosome and 1 Y chromosome, it is Carly male. Then this one, the CY, it is carrying both the recessive alleles. Okay, so obviously it will be lethal. Okay, got it? It will be dead. Now this one, see here very carefully. This one is carrying CY, so it is already dead for having recessive lethal alleles in homozygous condition, and along with that, it is also carrying L allele on its only X chromosome. Okay, so for both the reasons, this is lethal. Okay, it is dead. Okay. Next, CY plus, that means it is Carly and it has 2 X chromosomes, so Carly female. CY plus L and Y chromosome. So see here, though it has a Carly mutation in heterozygous condition, but as it is carrying the L allele on its single X chromosome, that is why it is lethal. So it is very important. So here lethality is caused due to lethal alleles on X chromosome. Okay, got it? Now see, CY plus, so it is Carly 2 X chromosome, Carly female. Then CY plus, Carly 1 X 1 Y chromosome, Carly male. CY plus, both the autosomes are carrying wild type of allele, so it is wild type fly with L plus, that means it is a female. Then here also both the autosomes are carrying CY plus, so it is wild type, and as it has 1 X 1 Y chromosome, it is wild type male. Then CY plus CY with L both X chromosomes, so it is a Carly female. CY plus with 1 X chromosome which has L allele, okay, lethal allele, and another one is Y chromosome. So it is also lethal due to presence of lethal allele or L allele on the sex chromosome or on the X chromosome of the male. Okay, got it? So here also male is dead due to presence of lethal allele. Here CY plus, so obviously it is wild type and 2 X chromosome, so wild type female. Here CY plus, but it has L allele or lethal allele on the X chromosome, so it is also dead due to what? Due to presence of lethal allele on the X chromosome. Okay. So let's see here. What was our given ratio? Our given ratio was how many Carly female is to male is to wild female is to male was there? 12 is to 6 is to 6 is to 3, which was given in our question. Okay, see here the ratio was given 12 is to 6 is to 6 is to 3. So the ratio is actually what? The ratio is actually 4 is to 2 is to 2 is to 1. Okay, and what we have derived from our checkerboard, that the ratio is again 4 is to 2 is to 2 is to 1. You can see here how many Carly females are there in our cross. How many Carly females? Let's count. This is one Carly female. Then where is second Carly female? This is second Carly female. Then this is the third Carly female. Then This is the fourth Carly female. So 4 Carly females are there. Okay. So see here, there are 4 Carly females. Then how many Carly males are there? See, let's check how many Carly males are there. See, this is the one Carly male, and then you can see this is the second Carly male. So 2 Carly males are there. So number is 2 here. Okay, got it. Then how many wild females are there? So let's check how many wild females are there. See here, this is one wild female, this is another wild female. So 2 wild females are there. See here, 2 wild females are there. And how many wild males are there? Okay, see, there is only single wild male. This is the single wild male. So it is 1. So you can see the given ratio in our question is matching with our derived cross ratio from F1 progeny data. Okay, so our cross is correct here. It is proven. Okay, now let's see. In this cross you can see total 16 progenies were formed. Okay, 16 progenies was formed, but from there 1, 2, 3, 4, 5, 6 and 7 are lethal, or 7 progenies are dead. Okay, already dead. And obviously then how many survives? Out of 16, when 7 are dead, obviously 9 survives, and you can see those 9 are formed by 4 Carly female, 2 Carly male, 2 wild female and 1 wild male. Okay, now see, among these 7 lethals you can see there are 2 lethal females. Okay, this one is one lethal female and this one is another lethal female. Okay, so there are only 2 lethal females, and those 2 died due to presence of CY. That means recessive lethal allele, Carly allele. Okay, they are not dead due to presence of lethal allele. Okay, see, because in this female the L plus was there in X chromosome, and in this female L plus L was there, so they are not dead due to presence of lethal alleles. Okay, because in one condition there was no lethal allele, and in another condition there was only one lethal allele, but as the lethal allele is recessive in nature, so it is dead due to presence of CY allele or recessive Carly allele, which is also a lethal allele. Okay, but L is a recessive Carly allele. So here as both the chromosomes, X chromosomes, are not carrying L, small l, so it is not dead due to presence of this small l allele, rather it is dead due to presence of this CY allele. Okay, so 2 out of 7 lethals, 2 are female whereas 5 are males. Okay, now among these 5s you can see that 2 males are dead due to presence of CY. Okay, so 2 males are dead due to presence of recessive lethal allele Carly in homozygous condition. Which one are those 2? Okay, first let me choose another one color from here so that I can show you. See here, this one male and this one male. Okay, these 2 are dead due to presence of CY allele or recessive lethal allele in homozygous condition. Okay, these 2 are dead due to that only. Okay, whereas though here you can also see that here L allele is also present, but first of all they are dead due to presence of recessive lethal Carly allele in homozygous condition. Okay, on the other hand you can see 2 males are dead due to presence of only lethal alleles on the X chromosome. Okay, and their genotype is CY plus CY, L and X chromosome, then CY plus CY with X chromosome carrying L allele and another Y chromosome. Okay, so see, where are those 2 males? See here, this male. Okay, this male has CY plus CY. Okay, that means it is not dead due to Carly mutation. It is dead due to presence of lethal allele on its single X chromosome. Got it? Now another one is, see here, this one. This is also a lethal male. Okay, this is also a male which is dead, and here you can see the CY and CY plus are in heterozygous condition, so it is not dead due to Carly allele, rather it is dead due to presence of lethal allele on its X chromosome. So these 2 are the Carly lethals. If it was not dead, it will be Carly in nature. Okay, so as it is dead, we can call it as Carli lethal, because here the Carli alleles are in heterozygous condition. Okay, so two males are dead due to two Carli males are dead due to presence of lethal allele on their X chromosome, whereas one male, which is wild type of male, was dead also due to presence of lethal allele on their X chromosome. And as it is a wild type male, so obviously its genotype is CY plus CY and then L and Y chromosome. So here you can see the last one, this male is actually a wild type of male because it has CY plus on both the X chromosome, on both the autosome. Okay, but it has L allele on its single X chromosome. So for presence of these L allele, it is lethal. Okay, got it? So these three, see here, these two Carli males and this one wild male, these three are dead due to presence of only L allele on their X chromosome. Okay, so you can now see that here there were 4 Carli females, so there should be 4 Carli male too, but 2 Carli males are dead due to presence of lethal alleles. Okay, got it? Whereas there was also 2 wild females, so there should be 2 wild males too, but there is only one wild male. Why? Because another wild male is dead due to presence of lethal allele. Okay, so that is how 50% of the males are dead in case of this question. Okay, got it? I think this is now clear to your mind. Now coming to another question, and this is the last question of today's video. See here, these are the F1 generation data with phenotype and their respective numbers, and you have to generate the P generation cross from this data. Okay, so first of all, let's see the sex ratio. The sex ratio is male is to female. How many males are there? 6 plus 6, 12 males are there. Whereas how many females are there? 12 plus 12, 24 females are there. So the sex ratio is 1:2. That means 50% of males are dead here. 50% male lethality cross will be there. Now let's discuss about the phenotypic ratio. The phenotypic ratio Carli is to wild is how many Carli winged flies are there? 12 plus 6, 18. How many wild types of flies are there? 12 plus 6, 18. So the phenotypic ratio is here 1:1. In our previous question, the phenotypic ratio was 2:1. Okay, so that was a monohybrid cross. But here the ratio is 1:1. So it is a test cross ratio. So in test cross, we know that one parent should be homozygous recessive. Okay, and here which allele is recessive? CY plus is a recessive allele. That means wild type allele is the recessive allele. Okay, over the mutant allele CY. So obviously one parent should carry this recessive allele in homozygous condition. So one parent must carry CY plus CY plus. Okay, and another parent should be heterozygote. That is how all the progeny genotypes can be formed. So in the first cross, we are taking mother as the homozygous recessive. So here mother is carrying CY plus CY plus, that is the homozygous recessive allele. Okay, and as it is a mother, so obviously it has 2 X chromosome, and we know that to do the lethality cross, the mother must have one L plus allele and another L allele. Okay. Whereas the father is here heterozygote, that is CY and CY plus, because in case of test cross, if you are taking mother as the homozygous recessive one, then the father should have heterozygous allele. Okay, CY, CY plus. And father have L plus on its X chromosome and a normal Y chromosome. Okay, I have discussed why there is L plus in my previous part. Okay, already. So what will be the gametes? Obviously from here only 2 gametes can be formed. One is CY plus L plus. Okay, and another should be CY plus L. Okay, so CY plus L. And from here 4 gametes can be formed. CY L plus. Okay, CY L plus. Then CY and Y chromosome. CY and Y chromosome. From here CY plus and L plus, CY plus and L plus, and another one is CY plus and Y chromosome. CY plus and Y chromosome. Okay. So when we are doing the cross, putting the female gametes on the upside and male gametes on the lateral side, we can see that 8 progenies could be formed. Here see the cross, 8 progenies could be formed. The first one is carrying cy plus cy, that means it is Carly, having 2 X chromosome, Carly female. Okay, the second one cy plus cy, so it is Carly with 2 X chromosomes, so it is Carly female. Then the third one cy plus cy, so Carly with 1 normal X chromosome and Y chromosome, Carly male. Then fourth one cy plus cy with 1 X chromosome having L allele and another Y chromosome. So it is a lethal male, okay, due to presence of L allele or lethal allele on its single X chromosome. So it is lethal, okay, it is a lethal male. Okay, so see here the male is getting dead here due to presence of lethal allele on its X chromosome. Then cy plus, both the chromosomes are carrying recessive alleles in homozygous condition. So it is wild type wing carrying Drosophila, and as it has 2 X chromosome, it is a female. Then cy plus, that means it is wild type with L plus and L, that means 2 X chromosome, so it is also a wild type female. Then cy plus with 1 X and 1 Y chromosome, it is a wild type female. Then cy plus with 1 X chromosome having lethal allele and another Y chromosome. So it is a wild male, okay, it was a Carly male, which was lethal, but here it is a wild male as it has cy plus, but as it has lethal allele on its X chromosome, it is lethal, it is dead. Okay, so these two are the lethal progenies or dead progenies from this cross. So in the question our given ratio was, what was our given ratio? See here from the question first. See, the ratio was 12 is to 6 is to 12 is to 6, right? The ratio was 12 is to 6 is to 12 is to 6. So given ratio of Carly female is to male is to wild female is to male was 12 is to 6 is to 12 is to 6. So it is 2 is to 1 is to 2 is to 1. And from our cross we have derived what ratio? Here from our cross we can also see there are 2 Carly females. See, these two are 2 Carly females. So Carly females are 2 in number. Then how many Carly males are there? There is only a single Carly male, so it is 1 in number. Then how many wild females are there? There are 2 wild females, this one and this one, so this is also 2 in number. Then how many wild males are there? There is only a single wild male, so it is 1 in number. So our ratio 2 is to 1 is to 2 is to 1 is matching with the given ratio 2 is to 1 is to 2 is to 1. So our cross is correct, it is proved. Okay, got it? Now, another cross can be also formed, which is known as reciprocal cross in case of test cross, where we will take the other parent as the homozygous recessive. Okay, see here we have taken female or mother as the homozygous recessive allele. Okay, cy plus. But here in the reciprocal cross we are taking father as cy plus or homozygous recessive here. Okay, with L plus allele on its X chromosome and Y chromosome. Okay. Whereas we know that in case of test cross, if one parent is homozygous recessive, another should be heterozygote. So here mother is cy plus cy with L plus and L, because L is needed for male lethality. Okay. So from mother we can get 4 types of gametes. One will be cy plus L plus, then another one should be cy plus L, then cy L plus, and another should be CYL. And father only 2 types of gametes, cy plus L plus and cy plus and Y chromosome. Okay. So when we will draw the checkerboard, keeping male gametes over here and female gametes over here, we will get cy plus cy, so it is wild type in nature, and 2 X chromosomes, so wild female. cy plus cy, wild with 1 X and Y chromosome, wild male. Then cy plus cy, it is also wild with 2 X chromosome, wild female. Then cy plus cy, but having 1 lethal allele on the single X chromosome, so it is a wild type lethal male. Okay, so here lethality has occurred. Okay, got it? Then cy plus cy, so it is Carly because it is present in heterozygous condition, and 2 X chromosome, so it is female. cy plus cy, it is Carly and as it has X and Y chromosome, it is male. CY plus CY, it is Carly with 2 X chromosome, it is female. Then CY plus CY, it is actually a Carly male, but as it has the lethal allele on its single X chromosome, this Carly male is lethal here. Okay, so this Carly male is lethal over here. So you can see two males, one is Carly and one is wild type male, is lethal here like the previous one cross. Okay, here also one Carly male was lethal and another wild male was lethal. Okay, so we know that the given ratio is 12:6:12:6, that is 2:1:2:1, and from this cross to this reciprocal cross to, we can also derive the ratio as 2:1:2:1. How? See here, how many Carly females are there? 2 Carly females. Okay, 2 Carly females are there, so it is matching. How many Carly males are there? See here, only 1 Carly male, so 2:1. Then how many wild females are there? 2 wild females are there. Okay, so it is also 2 in number. How many wild males are there? Only 1 wild male is there, so it is also 1 in the number. So it is matching with the given ratio in our question, so our cross is correct. Okay, so in case of test cross, you can see both the crosses are correct. You can do any one cross keeping only one parent as homozygous recessive and another one as heterozygous for the autosome. Okay, got it? And you have to put the lethality on the female X chromosome, 1 X chromosome, for getting 50% male lethals. Okay, so I think the video is clear to you now, and if you find this video useful, don't forget to hit the like button and share it with your friends, and don't forget to subscribe to my channel for more such videos. Thanks for watching and happy learning.